Hi, my name is Samuel Wang. I'm a second-year master's student in Dr. Hendricks' lab. Our lab is really interested in how cargoes, specific proteins, enzymes, and even organelles get to where they need to be inside the cell. And to answer those questions, we're interested in sort of the tracks that these cargoes take. So the microtubules, the proteins that decorate the microtubules, called microtubule-associated proteins, or MAPs for short, and of course, the motors that drive these cargoes to where they need to be. So our lab has many members from a wide variety of backgrounds. We have people from engineering, bioengineering, of course, but mechanical engineering, chemical engineering, and also from the more science side, biochemistry and neuroscience. Our team is very diverse, and each of us tackle this problem in a different angle, which really creates a very special environment inside the lab. Personally, I have a biochemistry background, and I bring that background by working with stem cells, because my project focuses on one of the MAPs, tau, that is only expressed in neurons. That's why I'm using stem cells, so that I can develop and differentiate human neurons and then study the tau protein inside these neurons. To answer some of these really important research questions, I think, like I said, a lot of people tackle the problem from different angles. So we have on the proteomic side with techniques such as Western blotting, mass spec, to sort of try to see the overall picture. What are the proteins associated with these cargoes? What are the motors? What are the adapters? What are the proteins, the MAPs that are on the microtubules? Our lab here has some pretty cool equipment. Our microscope here is outfitted with an optical trap, which is basically a focused beam. When this beam passes through a certain material, in our case we use beads that we internalize inside the cells. When this laser, so basically the laser traps this bead, and in our case, motors try to move the bead out of this laser, but this laser basically generates forces that basically pulls it towards the center. And at some point the motors can't compete with the laser's force, and basically detaches from the microtubule. And then we can measure a really important metric of the stall force of the motors. Another thing that our microscope has is TIRF, total internal reflection fluorescent microscopy, and basically this microscopy technique is really powerful to get a very small slice of the sample. And basically how we achieve that is, instead of having the laser emission straight up, which is epifluorescence, of course, we have it at a certain angle, and at a certain critical angle, we get a very, very clear image of at the sample plane. And with this, we can effectively eliminate a lot of background on the Z plane and have a really clear and beautiful image to make our measurements with. And this is used in many techniques, such as live cell imaging, that our lab does a lot. Live cell imaging is really powerful in our case because we're basically interested in sort of where the cargoes go, where they lead, how they get there. So with live cell imaging, with this technique, we can see these cargoes travel from either the periphery of the cell to the nucleus, the path they take, how much time it takes, of course, so the speed, does it switch directions, what's going on with these cargoes, and this technique really helps us with that. So this is our microscope. Currently it's encased in this box. This box is basically to keep in the temperature inside stable. We have a device that can heat up the ambient air to 37 degrees so that we can efficiently do live cell imaging over long periods of time. For the optical trap system, here we're in the back of the microscope where here this cord brings in infrared light. The optical trap uses infrared light. Here is a beam splitter which helps us control exactly how much light passes through. This device uses sound waves to change, ever so slightly change the laser's path. And basically here is a set of lenses and mirrors that guide the laser into the back of the microscope through this hole. The bioengineering department is spread across this whole floor, where most of the labs have their own little space, but a lot of the equipment that we use is actually shared among multiple labs. Here I'm going to be taking you to our common area where most people from our lab usually work. So this is our common area where we do most of our prep work. Here we have, of course, the glassware, where we store our reagents, where we have our water bath, and the centrifuge, the autoclave, the place where we do gels for Western blotting and gel electrophoresis. And here's where we do our cell culture. Our lab is equipped with biological safety cabinets that are shared in multiple labs. We have incubators. We have incubators over here where we keep our cells happy and living. I hope this virtual tour of our lab has been interesting and has inspired you to get involved into research like it did for me. One of the best ways to get involved with our lab is to apply for the SURE project.